So here we're going to come back to a concept that we've been discussing, and I want to try and reinforce in you these ideas of homologous genes, and then orthologs and paralogs, and then talk a little bit about how we can use these orthologs to construct phylogenetic trees. Now remember that these homologous genes are derived from a common ancestral gene. The orthologous genes, or orthologs for short, or the homologous genes are found in different species, whereas paralogs are ones that are found within a single species. We've talked about this concept of gene families. Those typically consist of two or more paralogs within the genome of a single organism. And the example that we discussed previously are the globins. There's also the interferons, are a gene family. There's many different types of gene families, and you could do a quick search to find a lot of relevant literature on these. Now, in terms of the alpha and beta globin polypeptides, the examples we're going to use are from humans and horses. In this figure, the way it's set up is what we have are the polypeptide sequences for the alpha, which are here in brown, and also the beta globins, which are here in yellow. So down here, the alpha and the beta. In the comparison, or these alignments, are these amino acid sequences for the human and for the horse. So what you can do is you can compare these amino acid sequences, and you can identify where there are differences. And these types of differences that usually arise are typically missense mutations. So for example, in just these first 30 amino acids, there are at least 4 differences, and these are all missense. Where's the second one? 15, 19, here's another one at 24. And they may be due to just a single nucleotide change, either at the first position. Second position changes usually result in really dramatic differences in these types of amino acids. But these are all missense mutations, essentially. Now, I've highlighted here the orthologs versus the paralogs. Remember, the orthologs are the homologous gene in different species. So we've got these two versions of the alpha. These are orthologs, one in the human, one in the horse. And then the second would be these two versions of the beta globin, one in the human and one in the horse. Whereas the paralogs are these alpha versus beta chains. Here outlined in red or highlighted in red are the paralogs in the horse, and then in black I bracketed the ones in humans. And you can look at the differences too there also. So this is just a short alignment. This is only 60. Yeah, a total of 60 for the beta and 55 for the alpha. You can see that there's some insertions in here that represent changes, dramatic changes in the nucleotide sequence for these. Now, when you start to align these and look at these differences, you can infer some patterns in evolution of these different types of these members of the gene family, of the globin gene family. And what it suggests by looking at this is that the gene duplications that created the alpha and beta globin genes occurred long before the evolutionary divergence of mammalian species. So there's been a greater amount of time for these alpha and beta globin genes to accumulate changes. So the duplication of that ancestral globin gene, if you remember, and I understand if you don't, is thought to have occurred approximately 400 million years ago. That would have been actually 200 million years before speciation of the mammals. So you had these duplication events occurring, and you've had lots and lots of time for these mutations to accumulate and for differences to arise. However, these genes are constrained in the sense that if you have too many changes within the alpha and beta globin, then you're going to sacrifice the ability of that individual to survive. So you can think of these as essential or vital genes This is all about transfer or movement of oxygen. So you don't want to be monkeying around with those sequences too much. So now let's look at these amino acid differences a little bit more closely. And I want you to notice the pattern here, that if you look at these orthologs, if you compare the human and the horse, we can, out of 141 amino acids, we can identify 18 amino acid differences. If you look at the beta globin, 146 amino acids total, we can see 25 amino acid differences. Now, if we compare the paralogs, remember that's the alpha versus the beta within a human, and the alpha and beta within a horse, you'll notice that there's a hell of a lot more amino acid differences between alpha and beta globin chains than what we saw in these orthologs between the human alpha and the human horse, for example. So the orthologs are more similar to each other than these paralogs are. And it's really even emphasized when you think about the size. These are relatively similar in terms of the number of amino acids present in those polypeptides, and yet you get these dramatically larger numbers of amino acid differences within a species. And that should make sense to you because alpha and beta globin chains are doing very different things, right? So there's very different polypeptides, different functions per se within that hemoglobin unit, whereas the human alpha has basically the same function as the horse alpha globin. Now, I don't want to walk away from this without emphasizing the idea again that these amino acid differences or the levels of differences that you see in these amino acids would be different than if you looked at the actual DNA sequences. So the DNA sequences could be have many more differences than what you would expect to see at this level.